Okay, so we are going to start chapter 10 today, and we're going to be going over DNA, RNA, and protein synthesis. So we're going to save the protein synthesis kind of toward the end of the chapter, and that's kind of the big idea and the big focus of this chapter. But we got to get to some things first. So if you remember not last chapter, chapter before last was all on mitosis and meiosis. Now, this was not discovered until the late 1800s. So keep that date in mind, late 1800s, almost the 1900s, all right? So right around that time, when we didn't know everything there was with mitosis and meiosis, there was a bunch of different scientists.
Seniors, may I have your attention?
Sorry, there was an announcement, but anyway. So they didn't know everything there was to know about mitosis and meiosis yet. We learned how cells reproduce. We were able to see some of the structures with prophase, metaphase, anaphase, telophase, but we didn't really know what was going on. So a few scientists did some different experiments. We're going to start off with Frederick Griffith's experiment. So what he studied was a strain of bacteria called Streptococcus pneumonia, and it actually causes pneumonia. All right, so when your lungs fill up with fluid, yeah, we don't want that. All right, so far, guys, pretty much we've talked about bacteria. We've talked about prokaryotic cells being bacteria cells, but we haven't talked about good bacteria and bad bacteria yet in this class. So we're going to talk about some good and some bad. When I'm talking about bad bacteria, I'm going to refer to it as the virulent strain of bacteria. So that's the bad one. Most bacteria is good. Most bacteria we want in our bodies, but this one we do not want. All right. So what he did, guys, is he had two different strains of bacteria. He had an S strain. That was the virulent one. Okay, the S strain is the bad one. So here's an easy way to think of it. S strain, S makes you sick. All right, S strain, S for sick. The R strain was the other one, and it's just normal bacteria. Nothing was bad about it. It was harmless. So what he did was he injected it into mice. Now, I'm just going to concentrate on these first two, and let's label them as to which one is which here. So we are going to start with and the mice are the same color, so I will refer to the bacteria. We will start with the blue bacteria over there. Now, if you look at the blue bacteria, they injected it, and the mouse was fine. So our first one over here, this is going to be our R strain. Okay? So that is the harmless one. Second one over here, they injected the mouse with the other bacteria, the red one, and sure enough, over some time, the mouse died. So this would be our S strain, okay? So that's all I want to do so far on this picture. We'll go through a little bit more later. So this was the first part, number 4. So he found that S strain killed the mouse, R strain kept the mouse alive. Now, before we go over number 5, I'm going to go back here. All right. In this one, the third one, what he did, and I always think of the movie The Water Boy. I don't know why, with Adam Sandler for this one. There's a scene in The Water Boy where he has a gigantic silver pot of water, and he has a flame underneath the water. And the one guy comes over to him, I think it was like the quarterback, and he goes, "Hey, water's better cold, buddy," you know? And Adam Sandler goes, "Oh, yeah, but I have the sterno here," and he doesn't really get the concept out, what he was trying to do. But what Adam Sandler was actually trying to do in that example is take all this water and produced high-quality water. So what he was doing was heating it to kill all of the bacteria that's in the water. moral of the story, guys, when we heat something, when we boil water, when you boil anything, you are killing the bacteria that is in it. Think of babies. Babies do not have a very good immune system when they come out of the womb. That's why you should always wash your hands around babies and things like that. But if you are giving the baby bottles, you always want to make sure you boil the bottles in boiling water. What that does is it kills any bacteria that could be in the bottles. All right. Now you don't want the milk in there, obviously. You don't want to boil the milk, but just the bottles you want to boil to kill it all. So what happened here in this third one is they had the S strain. So this still was the S strain. This still was the red one, but they boiled it. How they say in your book is they heat killed it. Okay. Pretty much they killed the bacteria. Now the bacteria is still there. It's just dead, So if they inject that into the mouse, it's still alive in the end. So that is our heat-killed one. All right? Whoops. So let's go to the fourth one here, the last one. This is the weird one. So here's what they did. I don't know what happened there. Hold on one second, guys. Okay, so I don't know why I'm in two locations, but we're just going to move me back over here because it's a little awkward. All right, so here's the last one. And here's what happened. So what they did was they took the R strain. So this is the R strain. The blue ones are the R strain. And they added it to the killed S strain. Oops. No, we'll just do STR for strain. So they put the R strain, which was harmless, with the heat-killed S strain, which also let the mouse live, but combined them together and the mouse died. So that's what we have to kind of wonder what happened. What happened to that last mouse that we put two things in that didn't kill it and it died? So here's your notes on that. The heat-killed S with the R strain produced a dead mouse. All right. So that's the one we got to worry about. So yeah, you guys would think that those two together would be fine. And you're like, no, Communal, you're wrong. In that previous picture there, no, he definitely should be alive. This picture is wrong. No, I'm not wrong. That is actually what happened. Let me explain to you guys what happened there. So, when the S strain was dead, so the actual bacteria was dead in the S strain because of the heat killing, we had the R strain, which was still alive. The S strain DNA that was dead actually transferred some of its DNA, which they called genetic material at the time because they didn't know about DNA. It transferred some of that genetic material to the alive R strain. And when it transferred it to the R strain, it actually made the R strain turn into S strain. So let me write that down for us. So the R strain turned into S strain. All right, which produced the mouse to be dead. All right. All right. Second experiment. This is Avery's experiment. All right. So what he did, oops, one too many. He tried to figure out if it was DNA, if it was RNA, or if it was the protein that transformed the R strain into the S strain in Griffith's experiment. So pretty much, guys, what he did was he took the previous experiment, and as I said, in Griffith's experiment, they just said genetic material. They didn't say DNA, they didn't say RNA, they didn't say if it was a protein. which that's the enzyme here. So this was the goal of Avery's experiment, to prove what one transformed the R strain into the S strain. So 3 different enzymes. We got this enzyme called protease. This destroys proteins. RNA-ase destroys RNA, and DNA-ase destroys DNA. So these are enzymes. Remember, enzymes always end in ASE. All right. Okay. So during the experiment, guys, like I said, he used these 3 different ones to destroy DNA, to destroy RNA, and to destroy the protein. Now, he did this in different mice. Okay, don't think he just did it in one mouse and put all 3 in. That wouldn't make much sense. So let me go over the picture and I'll go back to the thing. So in the first one, he destroyed the DNA. The mouse was alive still at the end. In the second one, he destroyed the RNA and the mouse was dead. Third one, this one is lipase. It destroys lipids. He destroyed lipids. The mouse was dead. Protease, that destroyed all the proteins. The mouse was dead. And he did a few other ones. Regardless of what he was doing, if he didn't destroy the DNA, the mouse died. So this made him conclude that DNA is the one responsible for transforming that R strain into S strain. If the DNA wasn't responsible for it, the mouse would still be dead. So in transforming the R strain to the S strain, that DNA was transferred from one to the other to make it virulent once again. All right. Okay. This is a virus. All right. Viruses are very simple. And I know coronavirus is like the big one right now, which is an RNA virus. But this one here, this is just a regular virus. We have a shell up top, and it's just a protein coat. That's all it is. And inside that like tan colored line there, that is the DNA. And that is pretty much all a virus is. So it doesn't have a cell membrane. It doesn't have organelles. It doesn't have the characteristics of life, which is why we don't consider viruses to be living. Now, this right here, guys, this thing, this is a cell. To be more specific, this is a bacterial cell. What can happen is this virus can actually almost like inject a needle into the bacterial cell and take its DNA and put it through the cell wall, through the cell membrane of the bacterial cell and get it inside the bacteria. All right. What this does is once the DNA is in the bacteria, the bacterial cell can become a virus-producing machine. And it just produces baby viruses. That's it. And the viruses develop. They get the DNA of this virus. And now we have a lot of viruses. This is how a disease, a viral disease, can spread in your body. It's going to go from cell to cell to cell. And those cells will be transformed to just make viruses. All right. So here is the virus. We call the different cells that it goes to, if they're bacterial cells, we call it a bacteriophage. But the virus latches onto the cell. You guys can see there, I don't have, well, the needle's in the last one, but once it latches onto the membrane, it can then put the needle into the cell through the cell wall and the cell membrane. If it's a bacterial cell, it'll inject the DNA, and then that DNA will become part of the bacterial cell genome. All right. So that is it for section 1, guys. Just the 2 experiments there and a little bit about bacteriophages and viruses. After this, so fast forward about 25 years. With our 2 experiments, with Griffith's and Avery's, we now know DNA is a transforming material. There was a big push to figure out the structure of DNA in the 1950s. Okay, so these 2 guys here, James Watson on the left here and Francis Crick, they were two of the scientists that were trying to define the structure of DNA. In the 1950s, they put it together. Took them a few years with the help of some other scientists and probably some stolen work. But they were able to see the structure of DNA. They were able to map it out. And they were able to see the DNA is in a helix form. This is Rosalind Franklin. Rosalind Franklin was one of the scientists that actually worked with DNA in a lab and did a lot of the research that Watson and Crick used to discover the structure of DNA. So what kind of stinks is she did a lot of the work, but she didn't get most of the credit back then. Now we do have a larger appreciation for Rosalind Franklin. But she worked in a lab with another scientist named Maurice Wilkins. She made crystals of DNA and used X-rays to be able to see these DNA crystals in a cross section of it. So this was like from above. Now, she died very, very, very young of cancer, probably because it was the 1950s. She was using X-rays all day long, probably didn't have lead bibs and stuff like that back then to protect herself. So she died extremely young of cancer. Maurice Wilkins gave this picture to Watson and Crick. And they were able to use that picture pretty much without her permission to come up with the structure of DNA. Maurice Wilkins, James Watson, and Francis Crick actually got a Nobel Prize for their discovery of the structure of DNA. Rosalind Franklin did not. Why? Because she had already died, unfortunately. So, yeah. All right. So those were the three men that got the Nobel Prize. Okay. So let's stop here, guys. For today, we will go over the structure of DNA with the nucleotides and everything that Watson and Crick published next time. Have a good rest of your day.